Hi, my name is Dr. Ed Park, and I'm recording the first of 9 lectures regarding exosomes and exosome therapy. In order to understand what exosomes are, we need to understand the endocytic pathway. The endocytic pathway is a broad term, meaning the stuff that goes on inside a cell, hence endocyte, and that's a pretty broad descriptor. As we can see from this cartoon, there are a lot of processes going on. First of all, we're making multiple little vesicles packaged in these bubbles, if you will. These are called MVEs, or multivesicular endocytic bodies, or endosomes. What's depicted on the right is the MVE excreting its content of multiple little spheres, sort of like dipping dots. But what's also depicted here to the upper right are microvesicles. In other words, the cell membrane can butt off for whatever reason, and they can also butt in. If we go further counterclockwise, we see what's a clathrin-coated pit, or CCV. This was the subject, I think, of a Nobel Prize awarded. In other words, there are proteins that are lying on the surface waiting to invaginate and create endocytes. So this is how the cell drinks in its environment, if you will. Now, what's depicted in the business end, or the center of the cell, is that proteins are being synthesized, they're being recycled. Sometimes the function of destroying toxins or repurposing them is performed in these lysosomes or peroxisomes. So the problem with describing things in terms of endocytosis is that we really just have a crude understanding. It appears that a lot of the business, both inside and outside the cell, is bound by membranes. So when we look at the 3 observable types of membrane-bound things floating in the serum, when the serum is where the cells float around, we have 3 gross descriptions. One are very large ones, relatively speaking. These are apoptotic bodies. Apoptotic bodies are just the remnants of a disintegrated cell. A cell that killed itself forms these blebs, and they're on the order of 1 to 5 microns, or millionths of a meter. Microvesicles are a broad category. They're hundreds of nanometers, and they bud off from the surface. What we're most interested in are these smaller spheres called exosomes. These are smaller than viruses, and they in fact were ignored for most of scientific history up until this point. They range in size from 30 to 150 nanometers, and generally they're very specific, and they contain specific surface markers as well as some DNA and lipids, but mainly we're interested in the messenger RNA, the proteins, and the microRNA, which are blockers of signals, as we'll explain shortly. So what isn't an exosome? Again, as shown in the last slide, the larger extracellular vesicles produced from budding of the cell, or even cell suicide, are not what we're generally calling exosomes. You can see in this scanning electron micrograph that you have a white blood cell attacking a damaged cell, presumably, and you see it disintegrating into a bunch of tiny blebs. The multivesicular bodies that we see again in this cartoon, that was reminiscent of the cartoon 2 slides earlier, there are multiple processes going on, and it's hard for us to discern how they interrelate or how they're separate. What is known from this study of Kowal et al. In PNAS is that when you spin them down, you get different densities, and the ones we're interested in, again, 30 to 150 nanometers. And in their study, they found 3 typical surface markers, at least in the populations that they were studying, that they're known as cellular differentiation 63, 81, and 9. So what is an exosome? Exosomes exist in all species, in all body fluids, and they're thought to mediate what we call the paracrine effect, or the way cells talk to and influence each other. Very rarely, although in some cases, especially with the immune system, there is actual physical contact and a handshake, but in most cases, cells talk to each other by sending out these exosomes. It should have been obvious, but it was ignored because we thought, well, there's proteins in the plasma or the serum, the fluid, so that must be how they talk. And all the stuff that was floating around was thought to be junk or debris. It was quite myopic on the part of scientists. And only in the last 4 decades have the occasional papers considered that maybe the junk that people were throwing out was actually the business end of what was going on. Again, as I mentioned, there are 40 to 140 nanometers around, and as I also mentioned, they are in these ILVs, intraluminal vesicles, or multivesicular bodies. I think the best way to visualize this is to look to the picture, the scanning electron micrograph, and you see MVB with 4 arrows. That is a multivesicular body with the tiny exosomes in them. And you can kind of infer that as it reaches the surface and it extrudes, you can see the exosomes coming off like Dippin' Dots or like clusters of grapes being released into the cytoplasm, where the exosomes will be protected from protein degradation and the sort of sea at large. In the last 12 years, there's been a hockey stick of research, especially in the last 3 or 4 years, although most of that is into cancer, because the exosomes from cancer are quite different. Other disease states are also being studied, and there's a lot of research going into using exosomes as drug deliveries. Think of it as a time-release capsule protected from degradation. So why do exosomes exist? Again, they mediate the paracrine intercellular communication. They help to reprogram other cells, and they really produce bespoke proteins, RNA, lipids, and surface receptors. So they're sort of like FedEx boxes filled with unique gifts inside. Contrast this with the apoptotic bodies, maybe some of the other microvesicles, which could be of mixed use or even trash. The song that's inside these tiny spheres depends on the musician, if you will, that's the cell, and the mood the cell is in other words, what it's responding to in terms of its internal and external milieu. But importantly, the dance that the song produces also depends on the listener. And we'll get into this a little bit in aging. If you're sending a young song to old cells, how will the cells dance? So what do exosomes do? Because of their size, they can go anywhere by any means. That includes blood, lymphatics, cerebrospinal fluid, even intracellular transport through the axons. The question is, are they address specific? And the presence of certain cell surface markers indicates there may be addressing going on, especially the ones from the immune system. Contrary to what some people believe, the ones from the immune system can have major histocompatibility antigens on them. They contain proteins, which would be like in the cartoon to the right, the accelerator gas. If you give a protein, it has an immediate effect. But they also have microRNA, which is like the brake. A microRNA is depicted on the bottom cartoon. It's a precise 22 base pair sequence that will match a messenger RNA. A messenger RNA is the message that will produce many, many proteins once it's fed into a ribosome. So a microRNA is a brake. It gums up on this particular sequence, so it has high specificity, and it will stop or prevent the translation. The microRNA cannot be fed through the ribosome. It cannot be translated, so it's a brake. And then finally, it has what we could consider, if you want to stretch the analogy, a clutch. In other words, the exosomes also contain messenger RNA, which messenger RNA can produce many proteins. So it's a way of like upshifting and increasing the speed of the gears. So instead of just one protein for one action or a break to stop the translation of certain messages you don't want made into proteins, the clutch or the messenger RNA is a force multiplier because it can have multiple uh, ribosomes making the same copy of the same protein over and over again simultaneously in what's called a polysome. So even across all species, these proteins, these messages, these silencing are respected. So when you drink cow milk, you're translating cow proteins, even though for all intents and purposes they're probably the same ones. And when they do experiments with mice, they use human exosomes because basically we have all the same machinery, and that's what's being made available currently in this very new area of study. This slide shows just some of the proteins from one of the company's exosome products. They assay them and they determine the concentration of different proteins. These proteins are things like growth factors, vascular endothelial growth factor, growth hormone, insulin-like growth factor, etc. And what we notice here in the green column, this is from amniotic fluid, and in the right is from mesenchymal stem cells. What we notice is that it's not a random distribution. Not every protein is represented, and there are orders of magnitude higher concentration in amniotic fluid as it results from a heterogeneous secretion from many cell types. The column to the right are the proteins from this one single mesenchymal stem cell from a single newborn baby, and they are quite different. So circling back again, just to remind you that a messenger RNA is like the clutch. It's a force multiplier. So what do I mean by that? On the upper left, we see the central dogma of molecular biology, which is that the DNA is unwrapped, it's translated into a message or a messenger RNA, and then by virtue of these ribosomes that run along the course of the messenger RNA, it prints out the protein like beads on a string. What's noted on the upper right is that multiple beads on the same string can produce multiple copies of the same protein as they run down the message. The histogram shows that these are some of the messenger RNAs that are found. Instead of the protein, these are sequences of messages that will translate into proteins. If you remember, the messenger RNA consists of a start and a stop, and in the middle are three letter, if you will, sequences that consist of RNA molecules. So the three-letter sequence denotes which protein, or excuse me, which amino acid will be assembled, and that's how the system works. What we see here is that the break, if you will, the microRNA, is a 22 base pair break that gums up a specific sequence. So if you have one message that you don't want translated, it will bind and gum it up, prevent it from going into the translation machine, prevent those polyribosomes from forming on the messenger RNA, and hinder this signal. The chip on the upper right is a Affymetrix product which measures the known human messenger RNAs. So if there's, well, it's an oversimplification, but we think there's about 16,000 human proteins with 16,000 messenger RNAs, although that's a gross simplification. Presumably you could tailor many combinations of microRNA to block all 16,000 messages. So are exosomes good or bad? People like asking questions like that. Well, they're neither good nor bad. It's just how the cells talk to each other and influence each other. Every cell makes them, so whether they're sending out trash or an important message or a dangerous message, it depends on what's going on. The packages determine the behavior. So if you look up this tiny URL that I created called WSCS18, that's tinyurl.com/WSCS18, WSCS 18, around mid-16. There's an amazing study by Dr. Li Chen who shows that if you give exosomes from a patient who's curing their fibrosis of the liver, it will cure other people developing liver fibrosis. And if you develop breast cancer, and another one of the lectures discusses that the exosomes from somebody who has got breast cancer that's spreading will cause transformation of benign tissue. So we're starting to understand that not only were exosomes important but ignored, but that the ecology of a system may be causing the cellular manifestations and tissue manifestations of disease. So if a cell is in crisis, perhaps that is a necessary and sufficient condition to make cancer or liver fibrosis. And these studies show that the serum containing the exosomes from cells and ecologies that have gotten over their problem can also be mitigating or even curing these disease states. So when we think about mesenchymal stem cell husbandry, if you will, or the growing of them, it's very important which cells we choose to produce our exosomes, and to borrow a wine term, the terroir is important. In other words, in winemaking, the climate, the soil, all these sort of conditions of growth determine how the grapes and the wine will turn out. So we also see that the conditions and the origin of the stem cells have vast importance in terms of what actions they'll produce in other cells. So as we wind up, we talk about exosomes as being songs, mixtures of notes, if you will, chords, progressions. This is a simple chart looking at the exosomes from different kinds of cells, ranging from immune cells, nerve cells, stem cells, and what we're seeing at first glance is this is a gross oversimplification. But what comes out of this is that, of course, depending on the function of the cell, what's put into the exosomes is vastly different. They have 16,000 choices, and especially in the case of immune cells, it's such a complicated endeavor that it's really hard for us to oversimplify. Yet oversimplify we do. This slide shows just a basic, and I mean that sincerely, a basic mock-up of how we understand some of the main pathways of biochemistry. Can you imagine with so many possible messages, so many possible proteins, so many possible blocking RNAs, how complicated the song is that these exosomes contain? So again, how do we know what the effect of exosomes will be? Biochemistry, usually we like to think of a single effect or maybe entourage effects, but we really are never going to understand how cells communicate the way that they do. Even single genes can never be said to have only one effect or even one variant. There's so much splicing, alternative folding. It's like saying, well, what does an F sharp represent? Well, in the key of C, it's a very dissonant sharp. But in Irving Berlin's favorite key, which he composed, it was the tonic or the root. So it all depends on context. In a concert, the effect of the molecules will depend on the way they're interpreted. In other words, the cells will dance according to their ability and their training as well. So what we can say is that if indeed we are all very similar to each other and to other animals, then mesenchymal stem cells tend to serve certain functions, and they include modulating inflammation, decreasing apoptosis, and promoting regeneration. And what's further, they do this not just by engraftment or differentiation, as was kind of favored, but indeed they send out exosomes. They are trophic, or they go to a region of inflammation, and then they start pumping out these little songs. Finally, why is there so much research into cancer exosomes? Well, cancer, of course, is a big business, and it's been known over the last, let's say, 8 to 10 years that the Exosomes from an established cancer ecology are different and they're dangerous. And in fact, this over 130 years ago was presumed or posited by a pathologist known as Paget. It's a lot of alliteration there. And to further, he called it his seeds and soil theory, which is that you can have cancer or precancer or whatever you want to call it, but if the soil is not ready, as Jesus said, then the seeds will not grow. So part of what we need to shift in our understanding of cancer and other diseases is that it's a failure of the ecology. The soil is prepped. It's made ready to experience the transformation that is so dangerous. The question of whether cancer exosomes are positive, negative, or mixed is very subjective. If you see exosomes from somebody who is in the midst of cancer versus somebody who has cured their cancer versus someone who is succumbing to their cancer, certainly we can guess that there's different degrees of success contained in the exosomes of that ecology. So I'll direct you to the fourth of the 9 lectures on cancer and exosomes where we describe this a little more in detail. Finally, how could exosomes be helpful? Well, in nature we know that mesenchymal stem cells are abundant in youth and depleted in old age. It's said that 1 in 10,000 cells in the bone marrow biopsy of a newborn is MSC, whereas when you're in your 70s, it's perhaps 1 in a million. So over the course of a lifetime, through mutation, silencing, ecology, epigenetics, depletion, or what we generally think of as aging, I suppose, you're running out of stem cell charge in your bone marrow. A robust mesenchymal stem cell recruitment and exosome secretion is typical when you have something like an injury, even states like cancer and autoimmunity. Perhaps the MSCs are there trying to orchestrate and direct and control what is going on. They seem to be sort of the quarterback and the coaches of our important processes of healing, of inflammation, and of the whole cancer autoimmunity axis. By preventing autoimmunity of the T cells, the exosomes also downregulate this important disease pathway. They also can encourage local stem cells to reproduce in times of injury. And then finally, by bringing in the later stages of what we call inflammation, they promote neovascularization or new vessels, and hopefully they strengthen the extracellular matrix. As we conclude, I just want to reiterate that exosomes are, in the case of what we're using, ethically sourced from a single C-section patient. The placenta of the newborn is donated and it's screened for infectious disease. And then from the Wharton's jelly of the cord come these mesenchymal stem cells. If you want to know more about the details of how this is done, see Lecture 6, please. The benefits of stem cells generally cluster around regeneration, resisting cell death, and rejuvenation. They have the upside of not encouraging graft versus host disease if they're not from immune cells. There are no self-antigens on the surface of MSC exosomes, so there's no graft versus host or host versus graft, which is when the immune system of the donated cells fights yours and vice versa. Another benefit is that they don't require engraftment. They are universally understood and accepted by all cells because we're all using the same toolkit. So you don't run into the same problems with destruction by your immune system, and also you don't run into problems with trapping and clumping in the lungs, spleen, and liver, which is common when you take another person's non-matched stem cells, or MSCs. If you're interested in learning more, I would go to one of the founders of MSc Science, Arnold Kaplan, and Google him or watch his videos on YouTube. In addition, if you want to learn more about exosomes, watch all 9 of the videos in this series, which will continue on as aging, and we talk about parabiosis, or mixing young and old animals. Lecture 4 is cancer, 5 is joints, number 6 is how these things are sourced, number 7, the FDA and that process, number 8, dangers, and number 9, what is my practice. So thank you very much for listening, and I hope to see you in the next lecture.